We've done our viruses, we've done our bacteria. Today we're going to do the same thing with the fungi, so fungal plant pathogens, and finish up after that with a couple of case studies. Fungi, fungal plant disease, are regarded as the most prevalent disease agent of plants. There's a lot more of them than anything else, maybe because they're also a lot easier to classify, but there's a lot more diversity. Bacterial taxonomists keep changing their taxonomy, so sometimes we have lots of species, sometimes we don't have so many because they've clumped and split them, but they're kind of all pretty similar. We've got quite a lot of fungal species, over 10,000. And they can obviously cause severe economic loss. So some of, if not the most important diseases of every major crop are related to fungal disease. And some classics, things like late blight in potatoes is obviously one of the classic plant diseases in terms of loss and impact on humanity. What is a fungus? Polyphyletic assembly of eukaryotic, heterotrophic, absorbative organisms that often develop rather diffuse, branched, tubular body and reproduce by means of spores. Gee, that's a mouthful. What does that all mean? Polyphyletic means it's actually from a number of phyla. So, classification of what is a true fungus and versus those things we kind of call fungi, but maybe really aren't fungi per se. But we'll call them that within this group of diseases of plants caused by fungal or fungal-like organisms. They're all eukaryotic. We'll talk about taxonomy, unfortunately, but we have to talk about taxonomy a bit later in the lecture, about the different phyla that these things belong to. They're all eukaryotes as opposed to our bacteria that are prokaryotes, as opposed to our viruses which are neither. All this sort of stuff is really about, they have that, what you've already seen in the lab, that mycelia, that hyphal structure, the mycelial structures, and generally reproduce by spores. But let's have a look at some of these things. So the fungal form, so that tubular structure, the hyphae, this thread-like structures we do see. So they're like little tubes. They may be multicellular, such as this. You can see those septa, those cross walls, as this tubular structures form. Sometimes they might be single cells. So certain species, certain genera don't have those septa, and so it's one big long single cell. But this thread-like tubular structures, a single strand is called a hypha. When you get a network, we call it a mycelium. So you've probably heard me say that in the lab class, and you'll read it in the textbooks. That's some of the definitions. So the hypha are the individual thread-like structures. When it's all as one big network, we talk about the mycelia present. So you might notice mycelia on the outside of an infected plant. You've got this network of hyphal threads. It generally has abundant branching, and sometimes that branching pattern is actually really important in trying to determine from a diagnostic perspective what type of fungus you've got. Often the hyphae and the mycelial structure or the hyphae are not terribly distinctive in terms of diagnostic perspective. They may have they will have different sizes. Sometimes they have distinct branching patterns, whether they're septate or not. Sometimes they have different colours and different growth rates on different media. That can be useful, but often it's the spores that are actually more important, or the spore structures, in terms of clearly identifying what we've got. So, what is a spore? That's the other major aspect. It's one or more cells, so they can be singular cellular, or they can be two or more cells in a single spore. It's analogous to the seed, I guess, of a plant. So this is the reproductive unit that is often used for disseminating the fungus from plant to plant or environment to environment. and allowing it to regrow. And again, a definition, a small, usually single-celled, but not always, sometimes multi-celled, reproductive body that's highly resistant to desiccation. So it's a survival structure. So it's, once again, very much like plant seeds. Plants grow quite happily. They produce seed to, one, disseminate, but also to survive. So they produce a seed that will actually overwinter, that will actually then germinate and start a new organism again. Much the same with the fungal spore. It's designed to try and disseminate. Some of them are short-lived for dissemination, and some of them are much longer-lived for survival. So often they are resistant to desiccation, heat, etc., and capable of growing to a new organism. And just some pictures. So you've got a huge variety of spores, and the actual structure can be important from a diagnostic perspective. So whether their shape, whether they're single-celled or multi-celled, like here, this is an open area with multiple cells. Sometimes like this, well again, sorry, this is an open area here with this club-like sort of shape. Some of them have rather interesting structures, but also even on the surface, whether if you look close enough, sometimes they'll have specific surface structures. So a lot of the rusts, for instance, will have spiky surfaces, but the actual surface structure can be important in terms of identifying specific species or genre. You have spores of different types, and really why is that? Because some are needed for dispersal and some are needed for survival. That's what we sort of mentioned before. So those that are designed for dispersal really are obviously adapted to move with whatever means that's available. So it might be airborne, it might be waterborne, in other ways to actually move either short or long distances to establish new infections. And some are designed to survive, so over winter, over season, so when the host has gone, it can hang around until a new host arrives and it can start that infection process all over again. And look, again, just a couple of examples. So here we go, Phytophthora, so Oomycetes. This is officially not a fungus by the true definition, but we call it part of the fungal family. So the Oomycetes. So it has these zoospores that are asexual zoospores. They're very short-lived. Some of those can perhaps survive only for hours or maybe days. They're very active. They swim through soil moisture and actively swim between plants and establish new infections. So important for dispersal, obviously not for survival because they're very short-lived. The alternate, the same organism, can produce these oospores, which are very thick cell walls. They don't move. They don't have flagella. They don't actually swim, but they survive. So they can survive for much longer term in the soil environment, for example, or associated with seed or some other structure, and survive in the longer term. Aspergillus and actinomycete, again, you've got three different types of spore types here. You've got this huel cell, which is once again a thick-walled survival structure. Doesn't really disperse terribly far. It's not what it's designed for. It's designed to survive. You've got the conidia, which are asexual spores that are relatively short-lived once again, but produced in massive numbers and tend to be wind-borne around. So once again, a dispersal unit, and an ascospore, which in this instance is similar to the huel. It's probably designed for survival rather than for dissemination. Rusts will actually have 5 different spore types, and they can all have different roles. So the teliospores for survival, the basidiospores for infection, for dissemination and infection. Spermatia, the male gametes for actually the reproductive process. The aciospores, again, short distance dispersal. and this is across different hosts. It's a complex relationship, which you might discuss later. And the arenospores, again, for dispersal over longer distance. So a bit tougher and can actually transmit over longer distance rather than short distance transmission. Short distance between, say, individual plants, longer distance, many kilometres. In terms of other important survival structures, you've got things that obviously those survival spores. You've got other structures that aren't strictly spores but are other survival structures. Chlamydospores, kind of called spores, I guess they kind of are, but they tend to be thickened mycelial or hyphal structures. And again, thickened, they're usually melanised, and survival structures. They'll actually enable them to overwinter in the environment they're in, whether it's in the soil environment or on a leaf surface or something. And there's some examples. Sclerotia are clearly designed for survival. These are melanised, hardened lumps of mycelia, so they're not spores at all. They're just an agglomeration of mycelia. They can be quite small, they can be quite large. That you can see that's the stem of a sunflower, I think. So that's probably about 8 millimetres across, 10 millimetres across. These things are 3-4 millimetres, so they can be quite large. These guys are much smaller. So these are highly melanised to survive ultraviolet light, and resistant to desiccation, etc. So other structures apart from spores can also be important for survival. So that's really basically structure of the physical structure of fungi, primarily made up of mycelia or hyphal structures forming a mycelial network that usually produce spores. Not every single fungus that we're going to deal with and every single fungal plant pathogen actually does produce spores. There are some that we call the imperfect fungi or that actually do not produce spores at all, and their entire infectivity process is through movement of mycelia, but most do. Anyway, a generalised pathogenicity cycle, much the same as what we had previously for some of these other pathogens. You start with the need to produce inoculum. So inoculum being the bit of the pathogen that's actually going to inoculate, going to come in contact with the plant itself. So usually that's a spore of some sort, but could be, as I said in some of those examples, could be germination of a sclerotia, so producing mycelia, infected mycelia. But usually it's a spore. So usually spores are produced, they then are deposited on a plant. Either it's on a leaf or on a root or something. That spore then, once it actually comes into contact with the plant, needs to germinate. So it's a bit like a seed, as we were saying before. It needs to actually wake up, germinate, and produce a germ tube. So that really is, most spores will, very much like a seed, they'll germinate and they'll start to produce a hyphal strand out from that spore, which may then start to form a specific infection structure. For some, but not all, fungal pathogens, they may form quite specialised infection structures. I'm going to talk about those in a minute, where they actually directly penetrate and interact with the host to sequester nutrients. Others will form other infection structures, whether they just directly penetrate through the plant with their hyphal structure and to start interacting with the plant itself. So they penetrate into the plant. Unlike, if you remember our viruses and our bacteria, they all needed help to get in. They couldn't actually penetrate through plants between plant cells or into plant cells themselves. Fungi, at least many fungi, have that capacity. They can penetrate through, they may still enter through natural openings, but they can penetrate into plant cells and force their way between plant cells as well in their infection process. So they germinate, they start producing a germ tube, they start producing the infection structures, they can then penetrate into the plant. So infection occurs, colonisation as the fungus grows and starts to spread. Unlike bacteria, again, where you've got one cell and make two cells and they're all kind of separate organisms, and you've got a collective population of cells. Here you've got a fungus that actually just keeps growing. So the network is connected generally, and so for each infection you've got one expanding organism, multicellular organism. Obviously then induction of symptoms, and then you start to, as they interact with the plant itself, so whether it's producing toxins again or enzymes or other disease initiating processes, then it needs to get from plant to plant. So it will generally produce spores or some other dissemination structure, and that will disperse from plant to plant. And then finally it needs to produce a survival structure to cope with interseasonality. So okay, the plant's dying, I don't want to die, I need to produce some, I need to have a strategy so I can survive between cropping systems or cropping cycles. So if we look at some of those aspects, for the spore germination, usually, for most spores at least, moisture is critical. So the presence of moisture will stimulate germination. So particularly those that deposit on, well, those in root systems too, but here an example of one where we're talking about spores depositing on a leaf surface. Just because the spores are inoculated onto a leaf surface doesn't mean that infection will occur, doesn't mean that germination will occur. Often there is a requirement for conducive conditions for a sufficient length of time to actually allow that germination and the infection to occur. So, a bit like, in fact, very analogous to a plant seed, the spore will obtain and uptake water, swell, allow them to start germinating, producing its germ tube, and then start the infection process. Temperature is also critical. They'll have temperature optima, so many diseases, for instance, will be, there are some that are encouraged by cool temperatures, some that are encouraged by slightly warmer temperatures, but there will be temperature optima that is ideal for germination and infection. Light, less so. And here's just an example of Botrytis cinerea. So probably that image we had at the very start. Where are we? That is probably brown rot, but it could be Botrytis cinerea as well. If that was a grey spores, that would be a very similar symptom to what this disease produces. Oops. In this instance, we're talking about in grapes, where we're getting the grey mould of grapes. So those, I don't know, has anyone ever had a botrytised wine? It's kind of a sweet wine with a slight funky taste. That's where they actually allow the grapes to be infected by this fungus, and this fungus shrivels the grapes up, so it actually concentrates the flavour. Adds a slight fungal taint to the flavour, but that's actually attractive in the final flavour profile. So you get this incredibly concentrated flavour because you've taken off a lot of the liquid from the grapes themselves as they're starting to shrivel and rot, and then they make a wine from that, and it's usually a dessert wine that's worth a fortune. Most of the times, however, they don't like grey mould because it actually is going to inhibit your normal wine production. And if you look at the types of environmental conditions that are necessary for grapevine infection, it tends to like temperatures around about 20 to 24 degrees, and it needs free moisture on the leaves or on the berries, should say, for at least, or ideally for 14 hours. And here's a sort of a graph where they've actually looked at that epidemiological profile. So leaf wetness duration, as you increase the duration of the leaf wetness, you increase the infection amount, and obviously there's an optima around that sort of temperature trial. So you've got a temperature by leaf wetness duration. So again, if we go back to our disease triangle for this particular example, if we don't have leaf wetness, or even if we have leaf wetness but only for 1 hour and then it dries up, infection's not going to happen. We've got a virulent pathogen, we've got a susceptible host, we might even have conducive environmental condition, but if it's not for long enough, if we don't have that long enough period, then infection won't occur. And why do we need that leaf wetness period? Because the leaf wetness lets the spore wake up, but then it needs to actually grow. It needs to produce a germ tube. It needs to then penetrate into the plant. And if it doesn't have sufficient time and moisture during that time to actually do that, infection won't occur, and you won't get disease. Infection. So, briefly mentioned that already. They can get in through natural openings, they can get in through wounds, but unlike the bacteria, unlike the viruses we talked about before, they can also, some of them anyway, enter by direct penetration, where they can force their way into a plant cell. In this instance, it's probably useful, just because we're going to talk about some of these highly specialised infection structures. If we talk about some of the different types of association with the host, we've got fungal plant pathogens that are obligate biotrophs. So these obligate biotrophs, these are the ones that have to live on a living host. They can't reproduce, they don't have a saprophytic lifestyle. They require to sequester all of their nutrients from a living host. So they don't kill plant cells. These are not necrotrophs. These are biotrophs. They actually keep the plant alive, and they suck nutrients from it. A bit like a vampire, isn't it, really? I don't know. Mind you, they do kill them, don't they? They just keep them semi-alive. No. So these guys here, they penetrate into cells and they actually absorb nutrients from the cells. Your necrotrophs, these are the ones that do kill plants and kill cells. They don't need specialised infection structures. They act in a similar manner to some extent to those bacterial pathogens, in that they will kill the cells and feed off the nutrients. They may be penetrating the cells as they go, unlike bacteria, but they will be producing toxins, enzymes, etc. that will kill cells in advance of their movement. And you've got some that are sort of halfway in between, that are hemibiotrophs that have the capacity to live off living tissues but don't have to. The difference between them, the obligate biotrophs produce a haustorium. So this haustorium is a specialised infection structure to absorb nutrients from living cells. And they can look like a whole bunch of different structures inside the plant cells. These other guys don't need it because they tend to be killing cells or sequestering nutrients in other manner. How does a haustoria work? So here's a rust spore from a Duromyces. Germinates, here's your germ tube. It produces an aprosorium, which is a specialised infection structure on the leaf surface. You get this penetration peg that forces its way through. And then you start getting this development, this mycelial development inside the leaf cells itself. So this is the intercellular stasis. Obviously here, these are the plant cells. But then it'll actually force its way into a plant cell and produce this haustorium where it'll interact with that host cell and sequester nutrients. So actually suck nutrients from that host cell. So here's it about to start into that particular one as well. So associates, punches a peg through, forms a haustorium and starts to sequester nutrients while keeping it alive. So that's a specialised structure as opposed to some of the other ones. Reproduction. So okay, we've got infection. How does it reproduce? Most pathogens will produce some form of reproductive structure within or on the surface of their infected plant tissue. And this is, from a diagnostic perspective, this can be really, really important, because here is when you examine your plant, your diseased plant sample, you've got your presence of your pathogen, but often also production of this reproductive structure, which often has quite characteristic forms or structures, particularly spore-forming structures, which can be very important in distinguishing one fungus from another fungus. So it might actually be the presence of mycelia, or in this instance mycelia with some of these conidial spores. Here's one example in vascular tissue, so you can see that the reproduction through the vascular tissue. This is going to lead to a wilting disease. Here's one where you've got a fungus that's actually infected this plant, but then it's producing this dissemination spore structure. So it's actually got this, what we call an asci, a spore. These are lots of fungal spores held inside this flask-shaped structure that the fungus has formed. It's put it on the leaf surface, that'll open up, and then the spores will be released. But that's a really nice characteristic structure. If you see that, you can actually then be fairly confident with the type of disease you're dealing with. Obviously, some of the basidiomycetes, they produce mushrooms, or in this case, bracket fungi, which again can be quite diagnostic in terms of the type of pathogen you're dealing. Rather than looking at a few mycelial strands, a few hyphal strands, you've got a fruiting structure as part of that reproduction, which is much more diagnostic. Now, fungi can also, unlike the viruses and the bacteria, which is somewhat more simple, they can have both both a sexual and an asexual stage in their life cycle. They don't all have to, but often they do. And terminology-wise, we can talk about holomorph, which is the whole fungus, and the anamorph is the asexual reproductive part. So often they might have an asexual reproduction where they'll produce conidial spores, which are just formed in a similar manner to the way bacterial cells split. You know, one cell splits into two, and so on, and just reproduces clonal material. But they may also have a teleomorph form, where two a male and a female version essentially of the fungus can mate and exchange genetic material and form a diploid, two-end structure and allow genetic exchange. So again, this is eukaryotic. This is important for the fungus in terms of its diversity and evolution. If it was completely asexual reproduction, it would have a limited genetic diversity and limited ability to adapt to changing environment. Because many of them have a sexual stage that allows them to be more genetically diverse, exchange genetic material, and adapt. So, for instance, a lot of, say, tolerance or resistance to fungicides that we might find in our plant pathogens, our plant pathogenic fungi, a lot of the development of that resistance is due to the capacity of that fungus to have a sexual reproductive stage, so it can actually adapt to a stress, in this instance a fungicide, and adapt and obtain resistance to that. So, for instance, late blight in potatoes, a nasty, horrible disease. It's not as nasty a horrible disease here in Australia, and our main reason for that is that we only have one mating type. So fungi aren't particular, you don't get a female fungus and a male fungus, but what you do get is distinct mating types. So you might get type A and a type B, and the type A and the type B can interact, but a type A and a type A can't. You know, they are similar, they aren't distinct mating types, they can't actually recognise one another and exchange information. So mating types are your equivalent of your capacity to interact. So with humans, we've got a male, we've got a female. That's how you can interact and allow sexual exchange. With fungi, you've got mating type 1, mating type 2, there might be mating type 3, 4, 5, and that allows that interaction. So with late light fungus, for instance, in Australia we have the A1 mating type, and that is all. So it can grow, it can cause disease, it can cause the disease symptom, but it can't mate. So therefore its genetic diversity is much reduced. And so fungicide control of late blight in Australia still works quite well with the standard fungicides. In Europe, in the US, in New Zealand I think as well, in Papua New Guinea, which is a bit scary because that's not far away, they have the A2 strain as well. And so there is sexual exchange happening, and in those countries you'll find fungicide control is difficult because it can adapt and overcome. It can generate greater diversity, genetic diversity, and overcome the capacity of the fungicide to actually work. Anyway, asexual reproduction. So your haploid or your single nuclei will divide by mitosis, and the progeny are sporangiospores, mitospores, or commonly we just call them conidiaspores. So a whole bunch of names for the same thing. So usually we talk about, with asexual production, you'll have structures and you'll have these conidiaspores. So you probably would have seen something, at least in the lab, things like, well, the example we had was penicillium. So it looked something like this. Here was your mycelia, then it had these structures a bit like this, and then a string of spores that were attached. So these are conidia, asexual spores, we call this conidiophores, which are the holding bits, the bits that hold on the conidia form from. So that's asexual reproduction with that particular fungus producing that spore type. So much the same as bacteria, where you got one cell split into another cell, you get clonal production. Sexual reproduction is where you get two haploids, so the two single copy of the nuclei, they fuse together to produce a diploid. So you actually get two, not diploid, you get two nuclei together. They then can interact and exchange genetic information, divide by meiosis, and you end up with these sexual spores that are 2N that can eventually divide back into single N. So, just important to note that fungi can reproduce both sexually and asexually. From a diagnostic perspective as well, and I think we cover a bit later, it gets kind of confusing too, because in the past at least, the asexual form of the fungus had one scientific name, and the sexual form of the fungus had a completely different scientific name, including a different genus. And it got quite confusing. It was the same organism, but had two completely different names because the actual morphological structures were quite different between the sexual form and the asexual form with those spores. That, thankfully, finally after hundreds of years, is starting to normalise, and they're saying we don't want two names for the same organism, so we're going to actually pick one and keep with that one name. So hopefully, certainly in the old literature, you'll find the same fungus called by two different scientific names, which is incredibly confusing. Confusing for everybody, even things like quarantine. I've seen examples where they say you can't bring that culture in because that's a plant pathogen and it's on our list, but you can bring that one in, which is exactly the same thing, but it's got the different name on it because it's not on our list, so that's fine. And you go, guys, it's the same thing. No. It's called different. It's a different name, so it must be different. It's kind of not. Anyway, disease induction. So how do we get disease? Similar to some of the things we talked about before, they may produce toxins or those biotrophs. They kind of penetrate and start sucking nutrients from the plant, so they're competing for resources. Others The necrotrophic ones may produce toxins, may also produce enzymes, often produce enzymes to help that penetration process in the first place anyway. They may cause physical damage by penetrating and punching their way through plant cells. They may block vascular tissue and lead to wilts, and there's a whole bunch of different means by which disease induction can occur. Most fungi, when they have to get from place to place, will move by spores, by means of spores, as we said before. And they can adapt to different means of moving from place to place. Many will spread be wind-blown, which is obviously efficient because they're very small, light things. You can blow them for quite some distance. Some will be spread with water, may even be spread through irrigation systems, or through soil water, or through rain splash. The rain just hitting it and blasting the spores away. But they may be associated with other biotic agents. Some associated with insects, birds, animals, humans, etc., to actually help move them around. They may also disseminate, but it's more a survival structure on things like the reproductive, seeds or other aspects of the host itself. Wind is probably the most common means of spreading most fungal pathogens. Mostly it's a passive process. It gets into the wind current, gets blown around, but not always. Some spores, some fungi can actually have active processes where to get it into the airstream, they'll actually shoot the spores into the air. They can forcibly eject them and get them into the air current and then blow them away. Obviously some of the ones that move in the soil moisture, like those zoospores we saw with the Phytophthora, they also actively swim in that soil moisture system. They have flagella, a bit like bacterial flagella, and they can actually swim through the soil moisture. They're just a hell of a lot bigger than a bacteria. And then they have to survive. So they can persist by producing these long-lived spores or sclerotia or other resting structures, as we talked about. Or they can persist by associating with the host, with seed, propagules. If it's a perennial host, they can just live in that infection site and wait for the new season to start and for infection to start again. They can live in alternate hosts, or alternatively they can live with plant debris. Those that have a saprophytic lifestyle, they might associate with fallen leaves, for example. So say like black spot of apples, that will commonly infect, it's a perennial tree, obviously. It'll infect the leaves, it'll infect the fruits. The infected leaves will fall onto the floor of the orchard, and they can sit there and reside there and wait till a new flush of leaves are formed, and then spores can be ejected back up and blown back up into the canopy and start a new infection. So again, in that plant debris at the base of the orchards. So for instance, in that system, one of the methods of controlling that disease, or one method that assists in controlling that disease, is actually removing of all the old leaves. So either blowing them away or even spraying urea across the base of the trees to encourage them to rot down much quicker. So by the time the new flush of leaves occur, the fungus is actually gone. Okay. Taxonomy. Yay! Or not. The last little bit of this lecture is really talking about classification, and from a diagnostic perspective that's obviously critical. If you don't know what you're dealing with, you can't devise a management strategy. So it is really quite critical. If we look at organisms in general, you can see they fall into 7 major kingdoms. You've got your archaebacteria, your true bacteria, plants, animals, and you've got these guys here that we tend to call, these are the true fungi, and these guys here are fungal-like things that will include plant pathogens. So when we're talking fungal plant pathogens, we're actually talking about organisms that sit in these 3 major groups. They are all eukaryotes and all have a nucleus. So out of those 3 groups, you've got your protozoan pathogens. So these really are more animal than fungal, I think. But traditionally have always been called fungi, and they do have fungal-like structures. So you've got your slime molds and your Plasmodiophorus that fit within that kingdom. Your water molds, your oomycetes within the Chromista. So again, they're not true fungi, so these are your Phytophthora, pretty damn important plant pathogens. Not true fungi, but we'll call them that for the sake of fungal plant pathology. And then you've got your true fungi, and you've got a whole bunch of different divisions within the true fungi. The chytrids, the zygomycetes, the glomeromycetes. These guys here that don't produce any spores at all. So these used to be called the imperfect fungi. The ascomycetes and the basidiomycetes. These are the ones that produce mushrooms. So we can argue that they're polyphyletic just because they go across multiple phyla with the plant pathogens. So there's a huge diversity of fungal. It's more than just genre. It's obviously fungal divisions, fungal kingdoms. So from a diagnostic perspective, how do we tell them apart? We can look at the nucleus state, whether it's a single N or 2N single nuclei, two nuclei type fungus, so whether it's the asexual sexual stage. Look at its cell wall composition, look at its basic morphology of the hyphae, whether the hyphae have septa, what sort of spores are formed, and how are they formed of the sexual and asexual. So from a morphological state, you can do that. Obviously, you can also use molecular tools to actually start looking, and more commonly these days, to start looking at the genetic diversity amongst them, and use some particularly conserved genes to actually allocate or to actually look at to see which particular species or genre or kingdom that the actual fungus belongs to. Now here's a table which I won't go over, but you can look at that if you want to. It's probably too small on this, but on Milo at least, you'll get a much bigger table to have a look at. But you'll see the different nucleus state, whether it's single N or 2N, whether it's hyphal-like fungus, or with these protozoan things, they actually don't have hyphae. They're more amoeba-like. So again, they're really more animal-like. What is the cell wall structure? Whether it's made of most of the true fungi are made of chitin. The oomycetes have quite different structures. So they're not they're not made of chitin. They've got a cellulose and glucan. Again, different. Whether it has septa or not, the true fungi do. The oomycetes do not. These guys don't have mycelia, so it doesn't matter. The type of asexual spores, these guys produce zoospores, so do these. These guys produce conidia. Sexual spores, you can get zoospore fusion. You get oospores, this sexual structure. Some of these don't have anamorphs. These are the imperfect ones, they don't actually have a sexual structure. And others have different types of structures. And again, whether they have survival structures, so whether they have sclerotia, chlamydospores, etc. So that's a crude example of some of the major groups. And if we can quickly go through some of those major groups before we get on to some case studies. So the Plasmodiophora mycota. These are these amoeba-like or really protozoan pathogens. They are all obligate plant pathogens. They all infect the roots. There's not a huge number of them, but they can be quite important. And one of our case studies will look at one of these. They produce zoospores to move between plants. So the zoospores have a pair of flagella. and they're formed in a structure called a zoosporangium. A zoosporangium, when you've got this angium, that's the structure that produces the type of spore. So a zoosporangium produces zoospores. These are released into the soil moisture. They swim around, find a root, start an infection, create a new zoosporangium, make more zoospores, and away they go in a polycyclic manner. The fungus itself, the term is a plasmodium. Once it's infected inside the root, and its survival structure, it'll produce either a cyst or a resting spore, which can allow it to survive. So these guys infect roots. They usually have a polycyclic infection process, and you'll get massive root infection. But then when the plant dies, it needs to survive because this is an obligate plant parasite, so it can't live saprophytically, it can't live on plant debris. So it goes and forms a resting spore, sits in the soil, and can survive for decades, often, waiting for a new host to come along, where it wakes up and starts the whole process all over again. Some examples. Is that it? Yeah. So here we go with our picture of a zoospore, a sporangia producing that. Here's our resting structure, our oospore. The imperfect fungi, so these guys don't have a sexual stage. These are common saprophytes, so they can live in dead and decaying material, as well as being pathogens. They're generally necrotrophs, kill things, but can also be facultative biotrophs, so sometimes they can live on living plants, but also can live on decaying material as well. They do not have a sexual stage, but can have a whole bunch of different types of asexual spores. So again, here's just some examples. Alternaria with a multicellular spore. Here's a Phyllophora with a single spore conidia. Here's our penicillium that I did a bad drawing of there, but that's what it looks like. An aspergillus, which looks a bit different, a little bit similar, a bit different, producing a chain of spores. They may also produce other structures. So here's a serverless with the structure on a leaf surface, so it kind of bursts open, and this releases all the conidial structures from the leaf surface. Or they might even have a pycnidia, which is a big fungal flask-like structure, which the surface here produces and releases all the conidia, and they kind of get released out through the flask end of the, or the mouth end of the flask. We can get ascomycetes within the true fungi, so part of the Euthymycota. Again, it can be saprophytes, facultative biotrophs, or some of them can also be obligate biotrophs, again a full range. These guys do have a sexual stage as well as an asexual stage. So the asexual stage can look very similar to the previous group. They might even have some that actually look very similar. But then they can have a sexual reproduction and produce a different form again. So usually identified by the sexual morphology, because the asexual morphology can look much like some of the other fungi. Taxonomy can get messy. So the ascocarp, the sexual morphology, is generally a structure, a epithecium, and that can produce and release these sexual spores. So again, these quite characteristic sort of structures. They may be completely closed, a clearthecia. They may have an opening, like that one there, which is a perithecia, or they may be cup-shaped, which is an apothecia. But they're all producing those sexual ascospores. I don't expect you to remember all the names of all these different spores. That's okay. The basidiomycetes, again, there's a range of different ones. They can be saprophytes, obligate biotrophs, or facultative biotrophs. They do have sexual and they do have a sexual stage. The asexual stage is more rare. You can have some that actually are what we call the sterile fungi, which don't actually produce any spores at all. So Rhizoctonia, which is a nasty pathogen of a whole range of things, will very, very seldom produce spores. There is a sexual stage that can be induced in some strains, but more often than not, you won't actually see any spores. So the diagnosis of these guys is all based on mycelial structures. You get those that produce mushrooms, they're Garrick's. Armillaria is probably the best plant pathogen example, so that's a tree pathogen. Some of them, again, these are the gilled mushrooms. These ones here are the paw-shaped mushrooms. So Ganoderma is another forest pathogen. And you get rust and smut, some of our obligate plant pathogens that you've seen some already in the prac class. The main diagnostic feature to know that you're dealing with a basidiomycete is in the mycelia. They actually form these clamp connections, so these actual structures where they form over and bind and actually allow that sexual crossing. So they can actually it's a cross where the septa forms. You get this little lump and this clamp connection, which actually allows the sexual exchange of nuclei from within the same mycelium. So nuclei A and nuclei B, which are distinct, but can actually then fuse to actually allow sexual exchange. Okay. So the take-home message is there's a hell of a lot of fungi out there, and there's a huge diversity, and they go across multiple phyla as well as multiple divisions as well as multiple species and genre. And there's an awful lot of them that interact with plants and cause disease across that diversity of range. So to try and understand what you're dealing with, you really do need to have at least an understanding of their taxonomy, of at least those major structures, and start to understand what some of those structures, associate some of those structures with a particular fungal groups. And then you can then start to devise management strategies based on what that actually means. Usually it's the spore-bearing structures, things like this, that are going to be most diagnostic. The penicillium mycelium or hypha is pretty much like lots of other hypha. This structure here, you see that, you know exactly what you're dealing with, apart from the fact you've got a green thing growing on your agar plate, which is also a bit of a telltale. In terms of management, you're really, once again, a bit like all the other pathogens we're talking about, you need to understand that life cycle and then understand when things are happening and why things are happening. So if we go back to that example with botrytis, grape rot, grey rot of grapes, you need to understand the requirement for, or when infections are going to occur, what stage the berry is going to be susceptible, what stage of ripeness. I'm trying to remember now. I think it's certainly not the green berries. As it starts to ripen, starts to get some sugars in that grape, that's when it becomes susceptible to infection. So you know there's a timing factor with the host. The host moves into a susceptibility phase. Then there's the requirement for leaf moisture or fruit moisture to actually allow that spores to germinate. Obviously there's a requirement for the pathogen to be present and to move through and deposit itself onto that fruit. And then infection can occur and disease will develop. Understanding those processes allow you to intervene in that process and maybe provide a critical intervention. And I can't overestimate that. Knowing the pathogen you're dealing with and knowing the host you're dealing with is not enough. You also got to understand that process. Probably a classic example that springs to mind is a disease that's actually less prominent now than it used to be, but a disease called ray blight of pyrethrum. So a nice local example. This is a fungus that actually invades in the flowers of pyrethrum. I don't know if pyrethrum is a daisy, and you harvest the flowers and you extract an insecticide from the flowers. So it's a natural insecticide you extract from the flowers. So obviously fungi that kill flowers is a bad thing. It's not what we want. So this was a flower pathogen. It invades the flowers, caused dieback of the flowers, and actually killed the flowers, and obviously killed your yield. It was identified relatively early by a bunch of agronomists that we think it might be this disease because we've read about it in chrysanthemum. Chrysanthemum's a related crop. Right. So let's look at the fungicide they used for controlling the chrysanthemum, and let's just toss it on. And they did that, and it didn't work at all. Why? Because they got their timing completely wrong. They didn't understand the biology of the pathogen. They knew what the pathogen was. They were absolutely right. They knew what the host was. They knew what the disease was. But they didn't understand when the infection was occurring. And by understanding that, all they needed to do was actually shift that fungicide program several months earlier, and they would have got control. And that's essentially what the industry has done. But understanding the life cycle is critical. Okay. Let's have a couple of minutes break. And we'll do some case studies.